Okay, let's look at some enzyme activity. So biological activity of an enzyme is dependent on maintaining its tertiary and quaternary structure. So the tertiary, remember, we have primary structure for these proteins. That's just the sequence of amino acids. And then they fold in on top of each other. You start getting some alpha helices and beta sheets. And then those structures fold in, and that's your tertiary structure. And then if you have two different polypeptide or polypeptide chains that interact, then that's the quaternary structure. And so in order for the enzyme to be active, you have to maintain that structure. You have to maintain the tertiary or quaternary structure. And since these are, most of these enzymes are proteins, they're made of amino acids. They're going to have those amino acid side chains that may be pH dependent. And so there's a pH optimum, and that's the pH at which the enzyme is the most active. And that really depends on the enzyme. If you have a digestive enzyme, then that pH optimum is going to be a lot lower. It's going to be under acidic conditions. Um, Most of the enzymes, non-digestive enzymes, but most of the other enzymes are going to have a pH optimum around 7. It's going to be around neutral pH. And that's why alkalosis and acidosis are potentially lethal. If your blood pH gets too acidic or too basic, then all your enzymes start to break down, all your proteins start to break down. They're not going to function anymore. They're going to unfold. And so if you can't maintain that structure, then then the protein's not going to work. It's not going to be in its biologically active form. If you remember what holds these proteins together, we have a lot of hydrogen bonds that hold like the secondary structure together. We might have some disulfide bonds in there. So once you start breaking all these things down, then the protein's not going to work anymore. Catalytic activity also increases with temperature up to a point. Up to a point, there is a temperature optimum, so that's when the temperature at which the enzyme is the most active. The rate's going to increase as the temperature goes up, right up to the point the protein starts to denature. So if you get too hot, then your proteins are not going to work any either. Your enzymes are going to start to unfold as well. So high fevers can denature proteins, then that can cause death. If the temperature is too low, then it affects the kinetic energy. Then that's not good either. So you're looking for that temperature optimum. That's where the temperature at which the enzyme is going to be most active. So which of the following won't affect the enzyme's biological activity? So we said temperature change. Temperature changes will affect things. pH, we said pH will affect things. And then of course if you need an organic compound and it's not there, if you need a cofactor coenzyme, you need that present as well. So the thing that doesn't really affect stuff is going to be the concentration changes, and we don't have to worry about that too much, but temperature, pressure, and the presence of organic compounds, that's going to be important. One interesting thing that I found in the book that they were talking about temperature resistant enzymes that are found in like these extremophiles. So in the hot springs at Yellowstone National Park, they found these really cool bacteria that have temperature resistant enzymes, and they were able to extract those and and now those particular enzymes that were found in that bacteria are used in PCR, so in polymerase chain reaction. When you do PCR, you need to have some of these enzymes that can withstand these high temperatures, and so now that's used in that technique in the lab, which is kind of cool. Catalytic mechanisms. So there are five catalytic mechanisms that we're going to talk about. The first one is electrostatic catalysis. So again, mechanisms, we're just looking at like how do these different enzymes work. So electrostatic, really we're looking at, so this green thing is the enzyme, this red one is the substrate, and the positive and negative, that's just showing the electrostatic interaction. That's just like a charge. So if you have one thing that's positive, it's going to be attracted to the negative end on the substrate, and the opposite over here. You can also have hydrogen bonding. Hydrogen bonding is one of the most common electrostatic interactions that are going to, basically you're just stabilizing this complex so that the reaction can happen. So you can have ionic, ion dipole, dipole dipole, hydrophobic, and hydrogen bonding. So any of those things where it's just based on like a charge holding the structure together, that's going to be electrostatic catalysis. Acid-base catalysis, when you have an acid-base reaction, when you have a hydrogen, a proton being transferred from one molecule to the other, that's an acid-base catalysis. And a lot of times that's combined with covalent catalysis. And covalent catalysis is when you're forming a covalent bond between the enzyme and the substrate, and that's what's holding it together. Strain distortion, this is often used in the induced fit model, so you may remember learning about that at some point. But you have your enzyme here, you have your substrate, you're kind of forcing it to occupy this particular space. And when you do that, everything lines up, and because of this distortion, strain it holds it in place, and then the reaction is able to happen. Proximity and orientation effects. This is when the enzyme bonds the reactants, the substrate, and orients them so that they are just within, really just putting them in the right place for the reactions to happen. So aligning the two reactants. So this one involves two reactants, of just holding both of those things in place so that they can react.